Hi, I'm Surag from Achal Lab at Stanford, and today I'll be presenting some of our work on dissecting the cis-regulatory landscape of somatic cell reprogramming from human fibroblasts to iPSCs. This is collaborative work done with the Blau and Wang Labs at Stanford, and all of the data has been collected by Mo. OX-OX, KLF, collectively known as Yamanaka factors, can be induced in somatic cells, for example, fibroblasts, and their induction begins a multiphasic cascade that culminates in the formation of pluripotent stem cell-like cells. This is, however, an inefficient process, and typically less than 1% of cells that you start with end up reprogramming successfully, and as a result, bulk assays tend to mask the diversity of reprogramming intermediates. One thing I want you to keep in mind is that reprogramming is not a linear process. And what I mean by that is that in addition to pluripotency associated regulatory elements that turn on either early or late during reprogramming, and somatic associated regulatory elements that turn off typically early during reprogramming, there's also a set of transient regulatory elements that stay accessible for different durations of time and in different intermediate cell populations, but are not accessible in the beginning of fibroblast or the terminal iPSC states. Mo collected 55,000 cells each from single cell RNA and single cell ATAC-seq from a 14-day time course sequenced at 2-day intervals. So starting with fibroblasts at day 0, we induced them with 4 sendai virus vectors, one for each of the 4 Yamanaka factors, and then sequenced these cells at 2-day intervals. And you can see that there are gradual transitions instead of discrete switches between cell states in these UMAPs. We can also computationally link cells between the RNA-seq and ATAC-seq modalities. And clustering in this case highlights 14 different cell states that are more or less concordant between the two modalities. These are the trajectories we obtained from our data. So in addition to a primary reprogramming arc of cells, we obtain a trajectory of cells that end up retaining their fibroblast-like characteristics through day 14. And we also observe the short-lived keratinocyte-like state, um, which mostly consists of cells from day 2. So next, we dissect the cis-regulatory landscape of reprogramming at multiple resolutions. And we begin with modules of enhancers that have correlated dynamics. So for example, I'm showing you modules of enhancers that are accessible in fibroblasts and close gradually over time and are close in pre-iPSCs. And an equivalent view of this would be by projecting the accessibility of modules of enhancers over individual cells in this UMAP. And so you can see that this UMAP represents an enhancer module that is open in fibroblasts and fibroblast-like cells and is closed in pre-iPSC-like cells. And so similarly, we can obtain modules of enhancers that are closed in fibroblasts, but gain accessibility either early or late during reprogramming. In addition, we see transient enhancer modules, many of which are specific to certain subsets of cell states. For example, this module of enhancers that is specific to the keratinocyte-like state, and another that's specific to the high-SQ cells, and also some cells along the primary reprogramming arc. And note that in typical bulk analyses, we can't distinguish between these peaks, sets of peaks, especially these transient peaks, as they're usually present in the same day. We can next link enhancers to the target genes on the basis of correlation between the peak's accessibility and the expression of the gene. And by doing so, we can associate our enhancer modules to modules of genes that have correlated expression dynamics and also obtain Go terms for each module. And so what we see here is that for peaks that gradually close over the course of reprogramming, end up being associated with genes whose expression decreases and goes down over reprogramming. And these are typically associated with fibroblast-specific terms like collagen formation, extracellular matrix assembly. Among the peaks that open up early or late, we also see an enrichment of reprogramming genes and Go terms, for example, regulation of stem cells, methylation, demethylation. To understand the cis-regulatory logic, at base resolution, we train neural networks inspired by the recent BPNet paper. For each of our cell states, we map a 2 KB input sequence using neural networks to the base resolution TN5 insertion profiles. I'm showing you a vignette at the Jun locus, with the observed reads in the fibroblast and iPSC cell states. And these are the model predictions for the same cell states on unseen sequence. You can see that the model predictions reasonably agree with the observed read profiles. We can then use methods like deep lift or deep shaft to interpret each of our models and retrieve cell state specific importance scores that highlight the motifs that are potentially regulating each of these dysregulatory elements in a cell state specific manner. And so we can use these concepts to interpret dynamic enhancers within loci of genes of our interest. And as an example, I'll be showing you the NANOG locus, which resides in this 200 kb subtag that shows gradual changes over the course of reprogramming the 3D architecture, but drastically changes in terms of its chromatin accessibility landscape, and in fact gains accessibility at multiple loci during reprogramming. The NANOG gene resides closer to one edge within this 200 kb region, and you can see that it gradually gains expression over the course of reprogramming. When we interpret these constitutive peaks at the edges, the model highlights CTCF motifs on both ends, which suggests that CTCF bookends this domain. Interestingly, this specific enhancer here, upstream of NANOG, seems to get activated before most other enhancers or peaks at this locus. And if we interpret this specific locus in a cell state specific manner with our models, we see a lot of dynamic activity. And zooming in, we can see that this site is littered with multiple OCT-SOX and KLF motifs within just 250 bases. And in fact, we can find OCT4 chip-seq peaks at the site in iPSC cell lines. And so this enhancer really seems to be the landing pad for the Yamanaka factors during the early stages of reprogramming. The nanopromoter itself seems to gain accessibility slightly after this enhancer. And again, interpreting this locus, we can see a very strong OCT-SOX motif that is bound and stays bound then till the end. In addition, we can see a peak that is gained somewhat around day 6 during reprogramming. And again, zooming in with our models, we can see that the model highlights yet another CTCF motif. And so a CTCF site is gained during reprogramming. And in fact, there is evidence for chips for CTCF being bound in H1ESCs and iPSC cell lines at this locus. And what's interesting is that if you look at the 3D chromatin architecture change between fibroblasts and ESCs, we see increased contacts within the region that is marked by this CTCF site. And so we can essentially repeat this process for any gene of our interest and dissect this dysregulatory logic at base resolution and focus on individual enhancers. An interesting phenomenon that we observe is what we term as anti-accessibility motifs. In addition to typical motifs that the model assigns positive importance scores to, we see multiple instances of motifs with negative importance scores. And these scores are negative in a context-specific manner because when we probe the same sequence with an iPSC BPNet model, we see that these negative scores are extinguished in these models. Upon aggregating these scores across the genome, we can see that it encodes the ZAP motif. And ZAP, interestingly, is a known transcriptional repressor. And more specifically, CDH1 is a known target site of ZAP repression. And so we can in silico delete either one or multiple instances of this motif at the site. And by doing so, the model predicts that in the fibroblast cell state, deleting all three instances of this motif would increase the accessibility by almost 2x. So that means it would almost double it, whereas it predicts almost no change in the pre-iPSC and iPSC cell states. More interestingly, when we eyeball these in a genome browser, we see that the ZAP motif instances tend to be farther away from the peak summit and in fact seem to be bookending accessible regions. And indeed, when we aggregate instances of these ZAP motifs and their distances from the closest peak summit, we see that in comparison to something like CDCF, which is enriched at the summit itself, ZAB instead is found farther away from the summit of peaks. And so this is something that's quite interesting and we will be validating soon. So finally, I also want to talk about how we've used our models to disentangle this relationship between OSK stoichiometry and motif affinity, which you get for free out of our single cell data. I'm showing you here a UMAP in which each cell is colored by a combinatorial state that depends on the expression of OX-SOX, KLF, and MYC. For example, fibroblasts here are colored in brown, which corresponds to cells that have low expression of all the four factors. And what I want you to note here is that our day 2 cells have extremely high expression levels of OX-SOX, KLF, and MYC, which gradually decrease over time into our pre-iPSC state cells, which have much lower levels of OX-SOX, KLF, and MYC expression. If you look at the primary reprogramming trajectory, here we're looking at the pseudotime analysis, and I've pointed out the main arrow of reprogramming in the attack and RNA UMAPs. If you look at the expression and also the ChromBar score, which is an indicator of motif activity, for the OCT motif, we can see that the OCT expression as well as the motif activity peaks at an intermediate time point and decreases further beyond that. And the same dynamics are reproduced for SOX and KLF. What this indicates is that a direct consequence of the expression of these factors being high early on in reprogramming is that OX and KLF seem to be promiscuously binding multiple peaks that are only transiently bound during reprogramming, which means that they're not accessible in the beginning nor in the terminal iPSC state. Now, even among transient peaks, we observe different modules of peaks that have different dynamics in terms of how long they remain accessible for. Now, I'm showing you peak sets 8, 10, and 11 here. And each of these peak sets gains accessibility at day 2, so as early as day 2, and are not actually accessible in the initial fibroblasts, but they differ in the duration for which these peaks stay accessible. So peak set 8, for example, is accessible only in this high OSK state, whereas peak set 11 stays accessible in more cells, in fact, stays accessible till about day 6 of reprogramming. And in contrast, we also have a peak set 13 that gains accessibility around day 2 and stays accessible for much longer up to day 10 and beyond. Interestingly, if we ask the question, how many of the peaks within each of our modules is captured in stable cell lines or tissues as assessed by the ENCODE DHS index, we find that most of our fibroblast specific peaks, for example, can be recovered in stable fibroblast cell lines and tissues in ENCODE, and the same holds for iPSC specific peaks. However, peak sets 8 and 10 specifically, peak sets that are transient for the shortest durations, uh, we find that a large fraction of these cannot be found in stable ENCODE cell lines or tissue types, and this suggests that these peaks are characteristic of reprogramming. And indeed, if we integrate our data with existing public ChIP-seq, day 2 ChIP-seq for OX, KLF, and MYC, for example, or the K4ME2 histone mark at different time points, we indeed see that our attack-seq dynamics can be recovered from other analysis of bulk reprogramming. And so this suggests that many of these transient peaks are reproducible features that are highly specific to reprogramming. And so we wanted to then understand how we could explain these peaks using motifs and different affinities of motifs. And so we compared the motifs that we extracted from these transient peaks to motifs extracted from peaks of iPSC cells. And so here I'm showing you the main motifs. In this case, those that were obtained from transient peaks versus those that were obtained from peaks in stable iPSC cell lines. And when we break these motifs down further into subclusters of motifs, you can see that there are subtle differences between these categories. In fact, the transient peak motifs end up being weaker instances of the canonical OX motif. So for example, if you look at the CAT here, which is recognized by the core binding domain of OX, while the CAT is more or less conserved in each of these subclusters derived from iPSC peaks, we see that some of the top subclusters in these transient peaks have an AAT instead of a CAT. And more interestingly, if you look at the flanks, the flanking TTTs, which is also recognized by the OCT protein, the flanks have much lower information content or have much lower frequency compared to the TTT in the case of the iPSC motif. And this hints at the fact that um, within these transient peaks, the motifs that are being bound are somewhat weaker instances than those that are bound by OX, KLF at much lower expression levels in stable iPSC cell lines. In fact, even between different transient peaks, so even between different transient peaks that are accessible for different durations of time, we can find a gradual shift in the motif strength going from peak set 8 through peak set 13. So what I'm showing you here are the log odds score, the motif log odds score of the instances of OCT-SOX motifs within each of these peak sets. And it suggests an upward trend where peak set 13, which stays on for much longer time, has stronger instances of the OCT-SOX motif compared to peak set 8, which is a short-lived state, and is dependent on extremely high expression levels of OCT-SOX and KLF. And this holds true not only for the OCT-SOX but also for the KLF motif, and we see a gradual increase in motif strength when we compare instances within peak set 13 versus peak set 8. So in conclusion, we are providing a resource for somatic cell reprogramming, and we're leveraging our cis-regulatory BPNet models to interpret chromatin dynamics at multiple scales. We can interpret individual bases, individual nucleotides and motifs within enhancers at base resolution in a cell type specific manner, and then aggregate this information over enhancer modules, and also um, in a genome-wide sense. Our models also highlight a novel class of anti-accessibility motifs, and we observed that these models can help us understand how motif affinity is associated with duration of transient peak accessibility. I'd like to close by thanking our collaborators, Kevin Wang and Helen Lab, who provided great assistance on this project, and Mo, who's collected all of the amazing data you saw in this presentation. And finally, my advisor, Anshul Kundache, for his support. Thank you. And with that, I'll take questions.